Good afternoon to all the NEET aspirants. This is your Prasanna ma'am from NEET World. So, here we are with the class Ascomycetes. We are done with fungal general characteristics and Phycomycetes. So, when we have to discuss the features of Ascomycetes, the very primary general characteristic that we have learned yesterday, that fungi shows great diversity in both morphology and habitat. If you have to look into Ascomycetes, this shows great diversity in morphology and habitat as we have discussed. Like usually Ascomycetes class has sac-like fungi. Unicellular fungi includes yeast to multicellular forms like Penicillium, Claviceps and Neurospora. As such, these will be the examples. So, they are from unicellular forms to multicellular forms. When you have to see the habitat of Ascomycetes, they can be saprophytic, decomposers, parasitic and coprophilous. Coprophilous are those fungi which inhibit dung. Fine, right? Those fungi which live on dung, they are coprophilous. And mycelium, if you take, we have septate mycelium in Ascomycetes, and what type of septa are there? Pore-like septa are present. Pore-like septa. Septate mycelium with pore-like septa. And next, if you see asexual reproduction. Asexual reproduction in Ascomycetes is by non-motile spores. Asexual reproduction is by non-motile spores. Asexual reproduction is by non-motile spores. Non-motile spores include conidia. Conidia are produced from specialized hyphae called as conidiophores. Conidia are produced from specialized hyphae called as conidiophores. Above conidiophores, some specialized structures are present, they are sterigmata, and over sterigmata haploid non-motile spores are produced, those are conidia. Conidia are the only exogenously released asexual spores, remember, they are the only exogenously released asexual spores. These conidia landing onto the suitable substratum will develop into a new mycelium. And apart from conidia, the non-motile asexual spores include oidea and sporangiospores. Sporangiospores. These are also non-motile and they are produced endogenously. Okay, non-motile and they are produced endogenously. This is about asexual reproduction of Ascomycetes. You have to remember the spores for sure, beta. Asked many times whether they are exogenously released or endogenously released. Here the fundamental thing you have to remember is exogenously released asexual spore, that is conidia in Ascomycetes, which is produced from a specialized hyphae, that is conidiophore, with the structures called as sterigmata. Clear, right ma? And then sexual reproduction is after the fusion, okay, fusion of male and female sex organs. Here the male sex organ is what? Antheridium. Female sex organ is Ascogonium. Post fusion, what happens? Post fusion, specialized hyphae are formed, those are ascocarps. And after fusion, diploid spores which are formed, diploid spores, or you can say diploid specialized cells, post fusion, they are called as ascus and the plural form is asci. And this is the site, ascus or asci is site for both karyogamy and meiosis. Karyogamy and meiosis. Fine. And this ascocarps, which are the fructifications that are formed, are of different structures again in different Ascomycetes. It can be cup or saucer shaped, then it is called as what? Apothecium. Flask shaped, perithecium. Globose shaped, clistothecium. Okay, cup or saucer shaped apothecium, flask shaped perithecium and globose shaped clistothecium. These are different shapes of the ascocarp. Clear, right? Everything is clear till here, right ma? Next, what are the examples of Ascomycetes? Important to remember the examples. I will give you a trick or mnemonic to remember the examples of Ascomycetes, that is A, C, P and T. Many at times I told you. Very simple example, wherein Aspergillus, Claviceps, Penicillium, Neurospora, Morels and Truffles and yeast. This is a simple mnemonic hack where you can easily remember the examples of Ascomycetes. See, Aspergillus, which is known as weed in the laboratory. Claviceps will cause what disease beta? Claviceps will cause ergot disease. Note down. Claviceps will cause ergot disease, and Penicillium neurospora, which is popularly known as Drosophila of plant kingdom. Drosophila of plant kingdom. Very important, note it down. Neurospora, which is popularly known as Drosophila of plant kingdom, and then yeast, morels and truffles, which are the edible delicacies. Okay, this is all about the class Ascomycetes. The next is Basidiomycetes. Basidiomycetes is said as advanced class of fungi. Okay, advanced class of fungi. Basidiomycetes is the advanced class of the kingdom fungi. It is called as advanced class because it has a very short-lived primary mycelium and a long-lived secondary mycelium. What is that primary mycelium? Primary mycelium is monokaryotic and short-lived. Okay, Basidiomycetes will have short-lived primary mycelium and which is monokaryotic. Okay, and the secondary mycelium which is formed, after plasmogamy, which is dikaryotic. Secondary mycelium is what ma? Dikaryotic. Dikaryotic secondary mycelium is long-lived. Dikaryotic secondary mycelium is long-lived. Fine. So, Basidiomycetes is the most advanced class of the kingdom fungi because it has a short-lived primary mycelium and long-lived secondary mycelium, and there are no asexual stages in the Basidiomycetes. No asexual reproduction or asexual stages in its life cycle. That is the reason it is called as the advanced class of fungi, and it is most commonly called as club fungi. Mostly the known forms of Basidiomycetes include mushrooms, puffballs and bracket fungi. These are the most known forms of Basidiomycetes, and then they grow in soil, on logs, in stumps, on logs, over tree stumps and in living bodies as parasites. Okay. The mycelium of Basidiomycetes is septate, and it has what type of septa? Dolichopore septal pores. Dolichopore septa. This dolichopore septa will have clamp connections like this. In between two septa, they have what? Clamp connections like this. This clamp connections will aid in equal distribution of dikaryon. They will aid in equal distribution of dikaryon. Fine, right? So, most of the mycelium is what? Secondary mycelium. Secondary mycelium is long-lived, which is a dikaryotic mycelium. Okay. So, the septal pores are what? Dolichopore septa is present, which has clamp connections. These clamp connections will aid in proper or equal distribution of dikaryons. Okay. So, I hope this is clear to you. Mycelium, what type of septa and how clamp connections are helpful. They are helpful in equal distribution of dikaryon structure. Next is reproduction. Vegetative mode of reproduction is of course fragmentation and all. Asexual reproduction is absent. Sexual reproduction is through what? Asexual reproduction is absent. Sexual reproduction is by sexual spores. Those are basidiospores. Basidiospores are exogenously produced from fruiting bodies. Those are basidiocarps. Exogenously produced from basidiocarps. Clear, right? Reproduction, vegetative, asexual and sexual. right. Next, sexual reproduction. So, here you can see after the fusion of male and female gametes, what happens is dikaryon structure is formed. Those dikaryon specialized cells are basidia. Singular is basidium, plural is basidia. So, here in this diagram you can see clearly where basidia are formed. They are exogenously present over the fruiting bodies, basidiocarps. They are present over the fruiting bodies, basidiocarps. Fine. These fructifications are also of many different structures from microscopic to macroscopic. So, here basidium, which is dikaryon structure or the diploid specialized cells, they are the sites of both. This is the site of both karyogamy and meiosis. Karyogamy and meiosis. Fine, right? Which is the site of both karyogamy and meiosis, and post this karyogamy and meiosis, haploid basidiospores are released. Basidiospores, which are the only exogenously released sexual spores. Okay, they are the only exogenously released sexual spores. Clear, right, ma? Clear till here? Yeah. Next, if you see, fructification is large and conspicuous in Basidiomycetes, and this is the most advanced class of fungi. I told you why is it the most advanced class of fungi. I hope the sexual reproduction and asexual reproduction is clear to everyone, ma. Now, if we have to look into the examples, agaricus, which is edible, which is mushroom. Eustylago is mud fungi. Puccinia is rust fungi. Okay. Polyporous, Lycoperdon, which are bracket fungi. So, agaricus, Eustylago, Puccinia, polyporous and Lycoperdon. How we can remember this? Polyporous, bracket fungi, polyporous, bracket fungi, Lycoperdon, puffballs. Okay. So, this you can remember it as a pulp. Fine, right, ma. Agaricus, Puccinia, Eustylago, Lycoperdon and polyporous. Okay. Next, if you see, yeah. So, these are the examples of Basidiomycetes. Next, the last class of fungi is Deuteromycetes. Deuteromycetes is the last class of fungi, which is fungi imperfecti or imperfect fungi, and the mycelium is again septate. Septate mycelium is present, and these are largely decomposers. If you have to see the habitat, these are largely what? Decomposers. Few of them are saprophytic and parasitic. As most of them are decomposers, they play major role in mineral cycling, and asexual reproduction here is again through conidia. Conidia, which are the asexual spores. What type of asexual spores? Exogenously released asexual spores. Exogenously released asexual spores. Clear, right? Next, this one. While the establishment of linkages, what happened is fungi with perfect sexual stages were moved again from Deuteromycetes to Basidiomycetes and Ascomycetes. Those fungi which are placed in Deuteromycetes, those which have perfect stages, perfect sexual stages, were again moved to Ascomycetes and Basidiomycetes. Okay, that is what which is explained in your NCERT. Do not get confused with those two lines or statements. Next, if you have to see the examples, Coletotrichum, Alternaria and Trichoderma, where you can easily remember it like cat. Okay. So, with this we have dealt with the complete classes of fungi. Now, in this table you can clearly predict or comparatively analyze the major features of all the 4 classes of fungi. Very important, ma, look into it carefully, very important. First, Phycomycetes is also called as what? Algal fungi. Algal fungi, and the mycelium is aseptate. Fine. Asexual spores of Phycomycetes include what? Zoospores, aplanospores, and sporangiospores. If you want, you can take a screenshot of it also, ma. Sexual reproduction or sexual spores include what? Oospores and zygospores. Fine. How you can remember the examples of this? I gave you an easy mnemonic, that is ram, rhizopus, albugo and mucor. Clear, right? Clear with the key features of Phycomycetes. Next is Ascomycetes, which is also known as sac fungi. Fine. Mycelium is septate. What type of septa is present? Pore-like septa is present. Asexual spores include what? Asexual spores include conidia majorly. Conidia and sporangiospores. Fine. And sexual spores include what, ma? Ascospores. Examples, major key examples include Aspergillus, Claviceps, Penicillium, Neurospora, Morels and Truffles. ACP and team, this is the mnemonic. Aspergillus, which is the weed in the laboratory. Claviceps, which causes ergot disease. Penicillium, from which the first antibiotic is produced, that is Penicillium notatum, from which penicillin is the antibiotic which is produced, right. Neurospora, which is popularly known as Drosophila of plant kingdom, and which is highly extensively used in biochemical and genetic works. Morels and truffles are the edible delicacies, and yeast, which is known as, mostly known as what? Widely known as baker's yeast or brewer's yeast, which is highly used in the daily bakeries and brewer's yeast in industries. Okay. Next, Basidiomycetes, which are puff balls, commonly called as what, ma? Puff balls. Commonly called as puff balls. And the mycelium is septate again. What type of septa are present, ma? What type of septa are present? Dolipore septa, which has clamp connections which aid in the equal distribution of dikaryotic nucleus. Next is asexual spores. Asexual stage is absent in Basidiomycetes. The only sexual spores which are exogenously released sexual spores include what? Basidiospores. Next is what? Examples. Examples include A pulp, Agaricus, Puccinia, Eustylago, Lycoperdon and Polyporus. Okay. Next is what, beta? Deuteromycetes. Deuteromycetes is called as fungi imperfecti. And mycelium is again septate and branched. Asexual spores include conidia, and sexual stages are absent in Deuteromycetes, that is why it is called as what? Imperfect fungi. Examples include Coletotrichum, Alternaria and Trichoderma. This is the complete comparative analysis of fungi. Please take a screenshot of this. Definitely you will get a question from this slide. Okay. Next, Kingdom Plantae and Animalia. Of course, we have two different chapters from Kingdom Plantae and Animalia, but then here we will learn in brief, okay, about both the kingdoms. Kingdom Plantae includes all the eukaryotic chlorophyll containing organisms. Cell wall of Kingdom Plantae is composed of what, ma? Cellulose, hemicellulose and pectin. Okay, and just most notedly we will call it cellulosic cell wall. Plant cell wall is cellulosic cell wall, but it is composed of cellulose, hemicellulose and pectin. Kingdom Plantae has 5 divisions. Kingdom Plantae is classified into 5 divisions. First is Thallophyta, which is algae. Thallophyta, which includes what, ma? Algae. Next is algae, Bryophyta, which are amphibians of plant kingdom. Third is Pteridophyta. First, terrestrial vascular cryptogams. Fourth is gymnosperms, which has snaked seeds. And fifth is the advanced division of plant kingdom, angiosperms. In all these 5 divisions of the kingdom Plantae, they exhibit alternation of generations. Alternation of generations which has haplontic phase, which is followed by diplontic phase. Or you can say alternation of generations. In between, one generation is haploid gametophytic generation and diploid sporophytic generation. Okay, haploid gametophytic generation which is followed by diploid sporophytic generation. Next, if you have to see nutrition. Nutrition is what mode of nutrition ma? Holophytic nutrition. Reserve food material in plants is what? Starch. Starch is the reserved food material. These are the basic key points actually, but then most of the students will make an errors in this as well. That is why pay keen attention and look into it ma. Few of the members in kingdom Plantae are partially heterotrophic rather than being holophytic. Few of the members of kingdom Plantae are what? Partially heterotrophic. They can be insectivorous plants or parasites. Insectivorous plants include bladderwort and Venus flytrap, and parasites include Cuscuta. Okay? Cuscuta is a complete parasite. Okay? Complete parasitic plants which dwell into both the xylem and phloem tissues of the preceding plant. It grabs both the minerals, nutrients from the plants. Okay? So, these are the partial heterotrophic plants from plant kingdom. Next is kingdom Animalia. It is generally described as heterotrophic eukaryotic multicellular organisms. Cell wall is absent. The limiting boundary of animal cell is cell membrane. We have learned that in Cell the Unit of Life. Nutrition is holozoic. What is holozoic mode of nutrition beta? The complex food material is ingested through mouth and that is digested to simple particles. Next, the reserve food material is glycogen. Reserve food material in kingdom Plantae is starch. Here the reserve food material is glycogen. Definite growth pattern is observed and locomotion is seen, and this has the organisms of kingdom Animalia has elaborate sensory and neuromotor mechanisms, and reproduction is sexual reproduction which is followed by the embryological development. Sexual reproduction is followed by embryological development. And then if you see those organisms which are not included in the 5 kingdom system of classification, which includes virus, viroids, prions and lichens. These are not included in 5 kingdom system of classification. First, if you have to see virus. Virus are called as infectious agents. These are non-cellular organisms. Virus are also described as nucleoprotein particles, or they are also said as just living entities, because they do not have a proper cell. They just has the genetic material which is covered by a protein coat, So, they are the infectious agents, non-cellular organisms. They are exceptions to cell theory as well. They are connecting links between non-living and living entities. Okay? Viruses are connecting links between non-living and living entities. Outside the living cell, they cannot replicate or they cannot divide. Virus cannot divide outside the living cell. They can exhibit the division or replication only inside the living cell or host. Now, let us see the discovery of viruses. Here 3 prominent scientists you have to remember. First is D.J. Ivanovsky. What did this scientist observe? tobacco mosaic plant which is infected, tobacco mosaic plant which is infected, he has taken the fluid of this plant and he has passed it through virus filter paper, okay, virus filter paper. He said that this fluid is being passed through virus filter paper, okay, and he is the first person to discover tobacco mosaic virus, okay. And then he said that the fluid is, the particles, the infectious agents pass through, infectious agents pass through bacterial filter paper. Second is W.M. Bajering, who gave us the concept of contagium vivum fluidum, contagium vivum fluidum, which means that those infectious agents which are passed through bacterial filter paper, they can infect the healthy plant as well. They have infected the healthy tobacco plant as well. So, as the fluid is infectious, he said it as what? Contagium vivum fluidum, and it is the scientist W.M. Stanley who said the crystallization of virus is possible. He crystallized viral particles. He crystallized viral particles. Clear, right? Clear till here, ma? Next. Here you see. So, virus consists of what? Virus consists of envelope, okay, envelope, protein coat and genetic material, okay. Virus consists of envelope, protein coat and genetic material. So, the envelope is made up of polysaccharides, lipids and proteins, any of these constituents. Envelope, viral envelope is made up of polysaccharides, proteins and lipids, any of these particles, fine. Example of enveloped viruses includes HIV or herpes virus, and then capsid. If you see, capsid coat has what? Capsomeres. These are arranged in helical, polyhedral and geometric forms. Capsomeres are arranged in what? Helical, polyhedral and geometric forms. Next, nucleoid, if you take, nucleoid consists either RNA or DNA. Nucleoid has either RNA or DNA. Animal infecting viruses has double stranded DNA. Animal infecting viruses has double stranded DNA. Examples include pox virus and herpes virus. Plant infecting viruses has single stranded RNA. Examples include TMV and HIV. Bacterial viruses are bacteriophages. Bacterial viruses or bacteriophages also has double stranded DNA. Till here it is clear, no ma? Nucleoid has also. Next, viral infections. In animals, usual viral infections that we come across include mumps, chickenpox, herpes, influenza and AIDS. And usual infections that we observe in plants include tobacco mosaic virus which cause tobacco mosaic disease, cauliflower mosaic virus which cause cauliflower mosaic disease. And then symptoms of viral infections, very important. Symptoms of viral infections include mosaic formation, leaf rolling, leaf curling or yellowing, vein clearing, dwarfing and stunted growth. These are the symptoms of viral infections. Most important, ma, please take a note of it or a screenshot of it. Next, importance that you have to learn: tobacco mosaic virus and bacteriophage. The structural description of tobacco mosaic virus asked n number of times in your EAPCET exam. Very important, please look into it. As we have learnt in the discoveries, tobacco mosaic virus is first discovered by whom? D.J. Ivanovsky. D.J. Ivanovsky has first observed tobacco mosaic virus. These are the elongated rod-like structures which are 3000 angstroms long. Okay, elongated rod-like structures which are 3000 angstroms long and 180 angstroms in diameter. Okay, 3000 angstroms long and 180 angstroms in diameter. They have a molecular weight of how much? Molecular weight of 39.4 into 10 to the power of 6 daltons, and they have capsomeres. Capsomeres are what? Protein subunits. Those are 2130 capsomeres which are helically arranged, and they have protein code called as capsid. And here if you see single stranded RNA, single stranded RNA is the genetic material which is present. It has how many nucleotides ma? 6400 nucleotides. When you have to take the ratio between the number of nucleotides and capsomere protein subunits, the ratio is what? 3:1 ratio. Number of nucleotides, that is 6400 nucleotides, is to amount of capsomeres, that is 3:1 ratio. Clear till here ma? Any doubt anywhere? Yeah. Next is bacteriophage. The genetic material bacteriophage has what? Double stranded DNA. It has tadpole structure. Tadpole structure will have a polyhedral head and a helical tail. Polyhedral head and a helical tail. Head is having double stranded DNA. Head is having double stranded DNA with neck and tail. Tail has helically arranged protein enzymes. Tail has what? Helically arranged protein enzymes, and tail fibers will end by tail fibers end has an enzyme called as what? Lysozymes. These lysozymes will degrade the wall layer of bacteria. Lysozymes will degrade the wall layer of what? Bacteria. So, double stranded DNA, it has tadpole structure, polyhedral head and a helical tail. Head is having double stranded DNA, it has neck, and tail has protein enzymes at the end like lysozyme at the tail fibers, which will degrade the wall layer of bacteria. Next, if you see viroids and prions, these are subviral agents. Viroids are those which has RNA without the protein coat. They just have single stranded RNA without protein coat. These are discovered by Theo Dionneer in the year 1971. This is called as virus without protein coat or virus with their coats off. Okay, viroids are viruses with their coats off, viruses without protein coats. They will cause what? Potato spindle tuber disease. Okay, viroids cause potato spindle tuber disease. They have single stranded RNA, single stranded RNA which tightly folded to form, single stranded RNA tightly folded to form what? Circular or linear structures. Okay, single stranded RNA which is tightly folded into circular or linear structures. This has a very low molecular weight. Okay, it has very low molecular weight when compared to virus. Next is prions. As viroids are those which has just RNA without the protein coat. Prions are without genetic material, they just has abnormally folded proteins. They just has what? Abnormally folded proteins. Nucleic acid is completely absent. They cause neurological diseases. They cause what? Neurological diseases. For example, scrape disease of sheep, mad cow disease in cattle, which is also called as bovine spongiform encephalopathy. Bovine spongiform encephalopathy. Encephalopathy. Bovine spongiform encephalopathy, which is commonly known as mad cow disease. And we have analogous variants of this disease also. Those analogous variants include CR-Jacob disease in humans. Okay, prions are clear, right? Abnormally folded proteins without nucleic acid. They cause neurological disorders. For example, scrape disease in sheep, mad cow disease in cattle. Another analogous variant that you see in human beings is CR-Jacob disease. Okay, see, these are the analogous variant. This is bovine prion, this is human prion protein. Both are prions only. If you have to see the molecular weight of prion, viroid and virus. Virus is of high molecular weight. After that, it is the single stranded RNA, right, the genetic material, after which the molecular weight is prion. So, prion's molecular weight is less. After prion it is viroid, and after viroid it is virus. So, if it is asked in the ascending order from less molecular weight to high molecular weight, high molecular weight. In the ascending order, so this is the direction. In the descending order, so this is the direction from virus to prion. Next is lichens. Lichens show symbiotic association between algal component and fungal component, symbiotic association between algal component and fungal component. So, algal component will prepare food and they will provide nutrition to fungi, fungal component will provide shelter to algae, right ma? Algal component will prepare food and they provide nutrition to fungi, fungal component will provide shelter to algae. This is the symbiotic association between algae and fungi which give out the structures of lichen, and lichens play a major role in they play major role as good pollution indicators. Lichens play major role as what? Pollution indicators, and they are most sensitive to air pollution, especially sulfur dioxide pollution. Okay, they do not grow in the polluted areas. Lichens do not grow in the polluted areas. With this, the complete concept of biological classification is done better. Now we will exercise all the EAPCET PYQs. Okay, so stay tuned for another 15 minutes where we will finish off the complete PYQs of EAPCET. Look at the first question. The question is open to everyone. Yeah, first question. Yeah. So, here you see puffballs belong to the following class. Which class? Puffballs? Basidiomycetes. Option C is the right answer. Second question you see assertion reason type of question. Organisms of Monera having pseudomurine cell walls produce biogas, methane. Yes, it is correct, but it is not all the organisms of Monera. Only Archaebacteria of kingdom Monera, Archaebacteria of kingdom Monera has what? They produce methane gas. Archaebacteria present in the gut of ruminants like cows, buffaloes produce methane. Yeah. So, here assertion is false, reason is true. Yes or no? Not all the organisms of kingdom Monera has pseudomurine in the cell wall. Eubacteria will have what? Peptidoglycan, not pseudomurine. It has murine in its cell wall, eubacteria. So, it is not all the organisms of kingdom Monera. In that case, assertion is false and reason is true. Next question. Match the following type of question, ma. Look into that and answer clearly. Yeah. Excuse. Answer clearly. Nostoc. Nostoc has specialized cells called as heterocyst. Okay. Mycoplasma causes, which is broom, which is pathogenic. Diatoms has specialized cells, ascospores. Sporozoan, plasmodia. So, A is 4, B is 3, C is 1, sorry, C is 2, D is 1. Clear, right? Yeah. Next, this one you see. Which of the following organisms is multicellular? Okay, yeast is unicellular, amoeba unicellular, bacillus unicellular, Spirogyra. Which of the following is multicellular? Spirogyra is multicellular, right? Which is filamentous algae. Next is what? Fifth question. Look into that and answer keenly. Character of organism from which the first antibiotic was discovered. First antibiotic is discovered from what? Penicillium. Penicillium. Penicillium belongs to which class of fungi? Ascomycetes. Like this you have to decode the question first, then you can analyze it easily. Character of the organism from which first antibiotic was discovered. First antibiotic was discovered from Penicillium, which belongs to the class Ascomycetes. This produces what? They has endogenously produced asexual spores. Endogenously produced asexual spores, what are they? Sorry, Ascomycetes has conidia, right? Exogenously produced sexual spores, endogenously produced asexual spores, presence of two nucleated cells during cell division. Yeah, option C is right. Presence of two nucleated cells during sexual reproduction, dikaryotic stage is observed. Dikaryotic stage. Okay. So, option C is right over here. Next, sixth question. Sixth question, you can answer. Which of the following show chromatophores? Chromatophores. Chromatophores are what? These are the invaginations of cell membrane into the cytoplasm which has pigments. This is observed in blue-green algae or cyanobacteria. These are the sites of photosynthesis as well. So, which of the following show chromatophores? Photosynthetic autotrophic eubacteria, yes, they are photosynthetic autotrophic eubacteria, they are not heterotrophic, they are not chemosynthetic, they are not branched filamentous bacteria. Photosynthetic autotrophic eubacteria are what? Blue-green algae. Option A is the right one. Seventh question you see, ma'am. Seventh question. Difference between holophytic and holozoic mode of nutrition. Holophytic and holozoic mode of nutrition. Holophytic is what? Autotrophic mode. Holozoic is ingestion of solid food or ingestion of complex food. Option A is right one now. Yes. Any objection, anyone? Yeah. Next question. Match the following type based on different characteristics and examples. This is how you have to decode and answer, which is asked in 2021 EAPCET. Okay. Coprophilus, whose habitat is what? Dung. So, here you can go with option 5, A is 5. Karyogamy and meiosis occur in sac-like structures. Those sac-like structures are what? Ascus. They are observed in ascomycetes. Here in these examples, what is ascomycetes example? Penicillium, right? Penicillium is the ascomycetes example. So, B you can take as 6, right? Next, flask-shaped fruiting bodies with apical opening. Flask-shaped fruiting bodies. We have seen cup-shaped, saucer-shaped and flask-shaped. So, flask-shaped fruiting bodies are what? Perithecium, which is option 4. Next, asexual reproduction takes place by aplanospores. Asexual reproduction by aplanospores, these are observed in Phycomycetes, Mucor, Rhizopus, albugo and Mucor. Excuse. So, option D is what? 2. Find right, ma. A5, B6, C4 and D2. This is the correct match. Next, this question. Excuse. Which among the following is not do not form asexual spores usually? Of course, the advanced class of fungi, that is Basidiomycetes, which is option B. Look at the 10th question, and in 10th question it is asked to identify the correct statements. No virus contains both DNA and RNA. Yes, no virus contains both DNA and RNA, either DNA or RNA. Viroids were named by Pasteur. No, viroids are named by T.O. Danier. Scrape disease in sheep is by Prions. 6 kingdom classification, 3 domains are present. In 6 kingdom classification, how many domains are present? 3 domains are present. So, virus does not contain both DNA, virus contains both, this is the statement actually, this is wrong. So, the right answer is option D. Next, match the following again. Cells with silica symmetrically rejuvenatory spores. Rejuvenatory spores you can see where, ma? In Chrysophyta, that is diatoms. Rejuvenatory spores or ascospores are observed. So, here A is 2. excuse. Next, holozoic nutrition, definite growth pattern. Holozoic nutrition, definite growth pattern and locomotion. This is observed in Animalia. So, B is 4. Zoospores, fusion of unequal gametes. Endogenous spores. Zoospores are heterocont usually. Zoospores have heterocont flagella. One is without any hair-like structures and which is whiplash flagella. One is whiplash flagella and other flagella has hair-like structures. This is tinsel flagella. Tinsel flagella. Zoospores, fusion of two unequal gametes, endogenous spores. These are observed in phycomycetes. So, C is option 3. Branched filamentous radial colonies, mycolic acid cell wall. This is observed where? In mycolic acid cell wall, phycomycetes 1. Okay. So, A2, B4, C3 and D1. Clear right now? Any doubts anyone? Next question, 13th question. 13th question. Answer everyone. Structure of blue-green algae that fix nitrogen. Direct question. Which are heterocysts, right? Heterocysts are the specialized structures which will fix nitrogen by what enzyme? Nitrogenase. Nitrogenase enzyme. This question match the following list. No sex organs, dikaryotic exogenous spores. Dikaryotic exogenous spores. Exogenous spores, basidiospores observed in the class Basidiomycetes. So, A is 4 here. Okay, A is 4. Infectious agents without a nucleic acid. Without a nucleic acid means they have abnormally folded proteins, which is prions. B is 5. Next, differentiated cytoplasm bloom in polluted water bodies. In polluted water bodies, blooms are formed by what? Trichodesmium erythraeum, okay, which is option 2. So, C is 2. Next, fruiting bodies beating spores survive many years. Fruiting bodies which survives many years. With beating spores, fruiting bodies which bear spores survive many years. Slime molds, right? Because slime molds will produce those spores which bear cell wall, which survive for many years. That is, D is option 3. Yeah. So, A4, B5, C2 and D3. Next, 15th question. Identify the organisms based on the given characters. Cell wall is pseudomurine. Cell wall with mucopeptide, marine bioluminescence. Cell wall is made up of protein rich pellicle found in stagnant water. Cell wall with pseudomurine is Archaebacteria. Archaebacteria. Cell wall with mucopeptide, Eubacteria. Bioluminescence, marine bioluminescence is Goniolax. Cell wall made up of protein rich pellicle found in stagnant water, which is Euglena. Fine. Next, assertion and reason. Assertion: the 6 kingdom classification by Carl Woese include bacteria, Archaebacteria, Protista, Fungi, Plantae and Animalia. Reason: if you see, Carl Woese divided 6 kingdoms into 2 domains. Now it is 3 domains. So, A is assertion is correct and reason is false. 17th question. Match the following type of question. Asexual reproduction. Asexual reproduction is by asexual spores, right ma? Which are by Conidia, exogenously released asexual spores, zoospores, motile asexual spores, sporangiospores, non-motile endogenously released asexual spores. Karyogamy, which is nothing but fusion of 2 nuclei, which is option 4. Meiosis, zygotes resulting in haploid spores by reductional division, that is meiosis, which is 5. Plasmogamy is fusion of protoplasms between 2 motile or non-motile gametes, which is option 1. This is the proper match between the column 1 and column 2. Next, 18th question, which is asked in EAPCET 2019. Identify the combination of incorrect statements related to cyanobacteria. Cyanobacteria is referred as blue-green algae, correct? They perform oxygenic photosynthesis in chloroplast, wrong. They perform oxygenic photosynthesis in thylakoid lamellae structures which are present in chromatophores. Chromatophores, where thylakoid lamellae structures. My God. So here chromatophores which has thylakoid lamellae, okay, thylakoid lamellae. In that what oxygenic photosynthesis happens. Next, red color of Red Sea by Trichodesmium erythraeum, correct. All cyanobacteria fix nitrogen in the heterosis, true. So the incorrect statement of cyanobacteria is what ma? Option 2. Next, assertion reason question in the euglenoids. Assertion, euglenoids have flexible body, yes, it is because of the structures called as myonims. In reason, they have protein rich pellicle which makes the body flexible. Yes, it is because of protein rich layer pellicle and by the structures of myonims. Okay. So, both A and R are true and R is the correct explanation of A here. Next, last question of today's session ma. Edible fruiting bodies called basidiocarps, they are produced by what? Edible fruiting bodies called basidiocarps. They are produced by Basidiomycetes. Amongst this, what is the Basidiomycetes? Agaricus. Puccinia, Claviceps, Saccharomyces belongs to what? Ascomycetes. With this, we have completely dealt what? Biological classification chapter. In our coming session, we will start with plant kingdom. So, stay tuned with the preparation of EAPCET 2026, highly useful sessions beta. I prefer this to all the TGE and AP EAPCET aspirants. Watch the sessions, for sure it is completely free in this public platform where the complete content and objective is delivered beta. Thank you and that is all for today's session.